this live streaming. I will edit the recordings later to remove the bit at the beginning, remove the bit at the end. And I will. So I'll write in the chat, live streaming at, so you can see that if you want to share that, Nicholas, with your parents. It will also pop up automatically on the London Futurist page if they were looking at that.
On the Facebook page?
No, on the YouTube.
Okay.
But if you give them that link, they will find it. Anybody who's subscribed to London Futurist will get the notification.
Okay.
And I'll put it on the meetup page as well. So that's there now. Good. And I'm going to press the button to start letting some attendees in.
Okay, great.
And now they'll come slowly.
Does that mean I start?
No, I'll let you know. We'll take a bit of time to get started.
Okay.
So, and usually, as I said, people will arrive after the top of the hour rather than before. But
yes,
we'll see how that pans out.
Mm.
In fact, nobody seems to be exactly waiting yet. Starting to arrive. No, it's starting to come. So welcome, Robert. Welcome, PNJ. And we'll just start once we've gone a few minutes past the top of the hour.
Sure.
Luiz. Ah, não, acho que era papi.
And
okay.
You sent that link?
Yes, I was able to send to my parents right now. In fact, I didn't anything. It was Nina with super skills with the cell phone here.
How good
As
are you?
I told you, we are very united. Okay.
Amazing. So your parents speak English as well as Portuguese?
My father speaks English. He liked very much your book, David, but
the one on politics.
The one on politics.
All right.
Yes, he asked me to tell this. My mother is a bit is not so good with English, but maybe she can catch some things, or my father can tell her. But I think they will. They are reading Harold's book together.
All right.
Yes. My father's English is better.
Are you going to translate the Harold's book, or is that a decision for later?
Well, it's a decision we have to make with Harold. And well, right now we were concentrated in the original English version.
Sure.
But I think if everything goes right and the research expand and everything else, maybe we could do versions in other languages, in Spanish, Portuguese, and other languages.
Well, if the research bears up its initial promise, more people are going to be saying, oh, I need to find out everything that I can about this. And they're going to be scrabbling for background information to understand not just the results, but what the meaning of the results is, and how it connects to the grand totals.
I hope it's going to be the next Origin of the Species.
Origin of the Species is a rather special book, of course. Yes.
Well,
Darwin had written many books before the Origin of the Species. I mean, this is your first book. And Darwin made a reputation for himself as a reporter for nature trips. You know, he went on this.
Yes, I've read Voyage of the Beagle. It's one of my favorite books.
So I've never read the origin of the species, but people say it's actually still very intelligible
Readable.
and cogent. Yes,
Yes,
even in the present day.
yes.
And he makes his case very carefully. You know, he points out, you know, there are all kinds of reasons why his theories make sense.
I mean, wait till my presentation.
All
I'm
right.
afraid I don't mistreat Darwin, but I'm afraid that several Brits have. And
Is this anticipating the discussion of group selection versus individual selection?
Yeah, for example.
Yes. Well, I think I'm with you on that. There is a sense in which group selection is a very real concept, despite people.
I mean, you know, are we on camera now? We're off camera. No matter.
Well, I'll remove this bit from the discussion later on. And by the way, if there's something you said at some stage and afterwards you think, oh, that was a bit unfortunate that you said something, let me know and I will remove it from the permanent record.
My knowledge, I never say anything but the truth, and I'm never afraid to say it.
Great.
Was about to say, in terms of group selection,
yes,
there's a very common phenomena where species replace each other, not individuals. Where, for instance, invading alien species like the snakehead fish invade a pond in New England. Even the biggest perch and the toughest sunny can't stand up the snakehead fish, and that's the way populations are really replaced. In the first oxygenation event on Earth, where photosynthesis started producing molecular oxygen, there was a huge group selection as anaerobes were forced to hide in the dark corners of the Earth, and aerobes came to the surface. So, I mean, it depends on your definition of group selection, but those are clearly cases in which groups have been selected, and usually by extraordinary and relatively common events, cataclysmic events, like volcanoes or meteors. Anyway, you know, Darwin himself never liked the idea that natural selection was all there was to evolution, and yet they've oh my gosh.
This might be
sorry about that.
A good time to put phones on mute.
Yeah, really good time.
Would
The heck? Okay.
Mission accomplished?
I hope.
Great. Well, I will kick things off in a moment. And so I'm going to stop sharing your screen temporarily, and you can put it back soon.
One sec, please. Oh my gosh. Sorry about all of this.
It's okay.
I never expected mutes. Let's so now it's on, and now it should be mute. Okay.
Excellent.
Okay, great.
So I'm going to kick off, and then I will. Was that another?
Just a message.
Just a message. That's fine.
I don't know why it's making this noise, and I can't turn it off. And I just got this phone, so I'm no expert on it. Maybe I should just put it in another room. Hold on a second.
Will do.
Wait, David.
Yes, sure.
We will mute our microphones, but in the start, you will present us. We would say
I think
something.
I will. Yes, you can just say Yeah,
Okay.
you're the publishers, and you will be in the background offering some comments from time to time.
Okay.
And let me get ready here. So let's get started. Welcome,
Okay.
everybody. Welcome, everybody. My name's David Wood, and I chair London Futurists. Today, we're going to be hearing about a possible paradigm shift in aging research, and our speaker is Dr. Harold Katcher, whose biography really defies any simple description. He has done lots of fascinating roles, and I think it's fair to say that the total of his work exceeds by some way the simple sum of its parts. So I'm not going to try and talk you through all the ins and outs, although Dr. Katcher himself may refer at times to some of the things he's done. It's possible, however, that what he's done in the last year or so will exceed by some way all the previous successes in his career. But in that case, as he will say, it's an extension of many of the ideas that he has been looking at. So I'm referring to the experiments involving the fraction of plasma known as E5, which we're going to be talking about in due course. I might mention that Dr. Katcher has admitted on camera recently he is 77 years old, which is one reason why he's had such a varied career. But I can also tell you that despite being 77, he is still very active. And I got an email from him the other day in which he apologized for not replying earlier to one of my questions. And he says, "I'm sorry, I've been working in the lab all day," which is an impressive accomplishment. And so there is still more to be heard. So before I hand over to Dr. Katcher, just a couple of words from me on this notion of paradigm shift. So I've discovered that both Dr. Katcher and myself have a fascination and an interest in the history and philosophy of science. And there is an argument that teaching people the history and philosophy of science might be almost as important as teaching them science. Of course, people need to know the best scientific current theories, but it's also good to understand what makes something a good scientific theory. And the notion of paradigm shift comes in there. This is one of the most famous paradigm shifts. This is how most people throughout most of history conceived of the motion of the heavenly spheres through the skies, with the Earth self-evidently being fixed and immobile at the center, with Mercury, Venus, the Sun, Mars, Jupiter, and Saturn, and others revolving around the Earth at the center of everything, a view that was amplified by statements in venerable ancient scripture. Of course, it was more complicated than this. The planets weren't just traveling in circles, they were traveling in circles within circles known as epicycles. From ancient times, at least some people were dissatisfied by that view. In the Islamic times and Arabs, there were some of their thinkers who disliked the idea, but it's mainly Nicholas Copernicus whose name is associated with this transformation. And he said, no, many things become clearer, many things become more straightforward once we put the sun at the center of our conceptual universe and recognize the Earth as just one of several planets revolving around the sun. We take this for granted nowadays, but for a while it was controversial. For one, because it wasn't actually as accurate as the old model, because the old model had these circles within circles, epicycles, and the predictions made straightforwardly by Copernicus's system were inaccurate until there were refinements to that initial revolution. And the refinements involved things like changing from circles to ellipses. And when that was done, the new paradigm went from strength to strength. Well, in a similar way, many of us futurists look at the possibility that there should be a Copernican revolution in how we look at aging and diseases. For most of history, people thought, well, there's all these things that can go wrong with the body, such as cancer, heart failures, dementia, stroke, diabetes. In earlier times, of course, lots of infectious diseases as well. And aging, oh yes, that's something else that tends to go wrong with the body, but it's left until last because it's a very natural thing, as natural as the Earth being visibly at the center of the universe, it seemed. And aging, therefore, received a very small share of funding. But the new Copernican revolution says, hang on, It's actually aging that should deserve most of our attention, because all these diseases are made more serious by aging. The older we are, the more likely we are to get cancer, and the more serious it is likely to be. So that's the second Copernican revolution. But as for the first, it needs more details before it actually becomes truly useful. And just saying aging is not enough. We need to answer the question, well, what is aging? And many futurists will probably say, well, aging is damaged molecules and damaged cells, which give rise over time to dysfunctional cell cultures, which damage the organs and later damage whole organ systems. And that is how the body comes to age and grow weak and feeble and eventually die. And if that's the whole truth, then where should we look to address aging? It's at this top level. We should intervene here to try to undo aging. But it's clear that isn't the whole story, because as well as molecules and cells being damaged, there are natural repair mechanisms in biology which often result in resetting the ages of cells and dealing in an effective way with damaged molecules. And when the body is young, these natural repair mechanisms are activated and supported. But when the body is old, somehow these mechanisms are hindered and they don't work well. When we see it like this, and this is something I've picked up from reading Dr. Katcher's book, maybe it's more useful to think, well, can we have a different characterization of what's different between a young body and an aging body? And perhaps that's where we should be intervening to undo aging. So I'm going to hand over in a moment to Dr. Katcher. But first, I want to mention one more thing, which is that we have another couple of people on this call too, Nicholas and Nina, who are briefly going to introduce themselves because they are the publishers of Harold's book, and they'll stay in the background, but from time to time they may offer some thoughts. So just introduce yourselves, please, Nicholas and Nina.
Hi everyone, my name is Nina.
I'm Nicholas.
And so we have a publishing house specialized in the rejuvenation field. We even translated David Wood's book, the Abolition of Aging, into Portuguese. And now we are the publisher of The Illusion of Knowledge by Dr. Harold Katcher.
That's great.
So, yeah, so we will be here in the background. So we maybe make occasional comments according to the presentation and the interview.
Thanks so much. So Harold, over to you. Okay.
To begin with, that was a very nice summary you gave and very clever. And exactly right. It's obvious to everyone who has eyes that the sun revolves around the earth every day. But and to suggest that the sun revolves, that the earth revolves around the sun and about its own axis certainly violates one of the basic laws of quote, science, what is it called? That the simplest explanation is the most likely one. Here
Occam's razor.
Occam's razor, thank you. This clearly violates Occam's razor, but it's the truth. The Earth does revolve around its axis, and it does revolve in elliptical orbits around the sun. And aging is not, I think the major problem with aging, I'll show you my presentation in but a second, but I think the major problem with aging research is that one of the major problems is that people are confusing cause and effect. But let's go. Let me start. Hopefully this is it. And I'll put it on slide view from the beginning and hope that this computer works. Oh, so far so good. Okay, well, this is great. That's a pre-made format, but a paradigm shift in the understanding of aging.
We're not seeing your slides yet, Harold. You need
Oh.
to finish the sharing. Maybe
Oh,
there's one more
pardon
button
me.
to press.
Oh,
It worked very well 15 minutes
it did,
ago.
It did. Well, hold on. Share screen.
Yeah,
If it
and then you
says so.
find the window.
Oh, I see. Okay.
And then you come down to the bottom again, and you.
And then I.
Fantastic. And then into the from the beginning.
From the beginning, and am I at? I can't read it now. I'll assume it's correct. We'll see in a second.
Click there.
And looks right.
Looking good.
Okay.
Over to you, Harold.
Sorry about that. Paradigm shift in the understanding of aging, and that's what we're talking about. The name of the book, The Illusion of Knowledge, comes from a quote by Daniel Borstein that's often attributed wrongly, but because he said it and agrees to it, Stephen Hawking. I also say it, and I also agree to it. You know, that the greatest enemy of knowledge is not ignorance, but the illusion of knowledge. That is believing false knowledge. It's quite a thing. Unlearn something that all authorities agree is truth than it is to learn something new. So pretty much because of my strange lifestyle and my not really caring about academic credentials, etc., and my rise to the top, which really happened anyway, I didn't care about what people thought. I only cared about the truth. And so let me tell you, kind of, first of all, where we begin. And this is where I began, with a complete belief in every one of these things. And what can I say? I taught them to my students. I used to teach the biology of aging. So aging results from the accumulation of damage at cellular and higher levels. Damage is the inevitable result of destructive, random, stochastic changes. Aging damage is irreparable, or at least there's no time to repair it, particularly damage to the genome, since that's the instructions for making everything else, either the nuclear or the mitochondrial DNA, for example. When damage accumulates to impede cellular and organismic functioning, this is called aging. So this is what most people call aging, an impaired and dysfunctional body, dysfunctional and impaired cells. This is what everybody believes pretty much, because there's a whole field called developmental biology, which seems to end at adulthood, which is organism development ends at adulthood when deterioration begins. So the assumption is that you reach adulthood, you're at your peak, and then you start to decline as stochastic damages accumulate. When deterioration becomes severe enough, death ensues, and organismal aging is ultimately the result of cellular aging, although that's logically, I should say, aging is death is logically the result of aging. But organismal aging is ultimately the result of cellular aging. When cells age, the tissues that those become less functional, and the organs that the tissues are part of become less functional, and the organ systems, etc. And ultimately the organism becomes dysfunctional, and we call that aging. And that's what most people believe, but I don't. Okay, so that all of those points are really what we would call a modern synthesis of aging. This is what most people, including David Sinclair, I believe, believes. but we'll see. This is poorly written. I meant to change it, but Darwin protested that evolution should not be reduced to natural selection, but his protests went unheard. And this is something I think you'd agree with. We know from the natural world that huge cataclysmic events and mass extinction actually accounted for much of the modern world. Okay. Later Darwinism. Now this is bound to be controversial. Sorry, but it's what I believe. I wrote the book for myself. Later Darwinism became a pseudo-religion, a secular religion opposed to Christians who attacked it. I think a lot of classical Darwinist attitudes resulted from an opposition to Christianity, an attempt to mechanize the world as just to rid it of supernatural forces. So I hear that the Christians, that a common cartoon I've seen in I don't know, London Times, a famous cartoon in the London Times, Darwin is a chimpanzee with Charles Darwin's face. You know, nobody would believe we came from such a lowly and disgusting creature as an ape. Although I don't see why anybody. Well, I'm not a great friend of apes anyway. Yeah, you can. The voice of authority. Yes. So the voice of authority, the people behind the Darwinist movement, I suppose you would say, reduced Darwinism to a mathematical certainty, a survival of the fittest, which I mention is actually a tautology, since the fittest mean those most likely to reproduce or produce reproductive offspring, right? So saying that the fittest will be more represented in the next generation. This is tautological, which means it gives no actual information if fittest means just that. Now I say the classical pseudoscience that calls itself evolutionary theory further shackled itself by not permitting group selection, the idea that selection could occur between groups or species rather than between individuals. This made the concept of an individual decreasing its lifespan for the good of the species a real possibility, because continued lifespan meant continued reproduction. Continued reproduction meant more offspring. All more offspring meant fitter. Okay. And the exception is, of course, the social insects, where the queen provides the genetics for the entire hive. In the late 1800s or early 1900s, August Weismann, who is regarded as one of the greatest biologists of his time, proposed that organisms, in order to make room for their evolutionarily advanced offspring, because their offspring have had one more round of selection than they have had parents, right? You understand? So there ought to be a death program to eliminate the parents, which would have, significant advantages over the offspring in terms of, for instance, knowledge or size or sheer numbers. There ought to be a death program to eliminate the parents so that the more evolved offspring replace them. And this is biology's answer to variation. nobody liked this idea at all. The idea that we are born to die, that a death program could exist, it was hateful to most biologists. Personally, I think for psychological or religious reasons, or anti-religious reasons, rather than good science. I think if you look straight on at the obvious fact that species have maximal lifespans, you know, that a dog will live to 12, 14, 16, 18, 20, but he won't live to 25, and he won't live to 30, and he won't live to 40 or 50 or 60. And a human has his own maximum, which they say is about 120 years. So far, the highest recorded age, I think it was Jean Calmet, was 122 let's about as long as human beings live. And let's continue with the next slide, and I'll make my points there. So here it says that most scientists studying the biology of aging believe one to all of the following authorities in their quote theories. Those are what are popularly known as sneer quotes. Yeah, I mean to be controversial. I don't because it really bothers me. A theory is normally the result of a bunch of proven, quote proven, because you can't prove a hypothesis. An hypothesis, you can only disprove one, but hypotheses that have never been disproven, that explain in general a large phenomenon like aging. Real scientific theory should be able to make prediction, and none of these quote theories that even claim to make predictions. All right, so let's go through what everyone believes. First is Peter Medawar. He was a brilliant transplant. He was actually the father of transplantation. So he was a brilliant surgeon and a scientist. And he, upon discovering that as animals age, the reproductive rate decreases, came up with a theory to explain aging, in that mutations that will only appear in old age tend to accumulate because there's no selective force against them, since reproduction has slowed down at old age, and a gene manifests past production will have no effect on whether or not the organism reduces or not. But that involves circular reasoning, because then you have to ask yourself, well, why does the organism's reproductive rate taper off as it ages? By the way, that's not true in many cases, and naturalists have shown that that's not the case. But let's assume that it were the case. The only possible cause for that would be some sort of aging phenomena that slowed down reproduction or stopped reproduction. So now you have to say, well, what's responsible for that phenomenon? And you'd have to implicate aging genes, which effectively is circular reasoning. You're saying that basically aging is the cause of aging. And that's it. There's, again, no information on that. It sounds very clever. It basically means that mutations that affect aging and accumulate totally randomly determine the age that a creature will reach. There's actually no mechanism for why these genes will suddenly assert themselves at older ages, for example. Anyway, it doesn't make sense, and it doesn't explain the facts. I'll tell you what the facts are in a bit. Okay. Next, there comes George Williams. And he came up with a theory, which I never wrote down, called antagonistic pleiotropy. Pleiotropy is the property of at that time, this is 1957, that time recently discovered a fact that one protein, one function is not in any way nearly true. Almost every protein has multiple functions, is involved in multiple systems. Sometimes the functions are bizarrely, almost scary, that a one-dimensional string of amino acids can do such a thing, of course, by folding itself into a three-dimensional machine. An enzyme involved in the breakdown of sugar, when it loses the sugar that it breaks down, will actually go to the nucleus, become what's called a transcription factor, and cause the cell to take in more of that sugar. So, I mean, this is a single protein, an unintelligent thing you can write down the structure of on a piece of paper, and it'll do these amazing things. Anyway, George Williams said that a protein that would have a pleiotropic form, a Dr. Jekyll form, if you would, during youth, so that it encourages or increases the ability to reproduce, might then have a pleiotropic form that during aging actually is deleterious, actually harms the animal. But it doesn't matter, because that harmful form occurs after the reproductive period. So it's totally protected. And the form that enhances reproduction is selected for, not selected against, even though it causes aging and death. My biggest problem with this is that I don't believe there are such proteins. They give examples. If you read my book, I explain why the examples they give are totally fallacious and actually don't prove the opposite point. In fact, what I write is okay. It assumes that genes act deleteriously, and then I have switch genes because I cannot name any. They talk about mTOR. I go into it in the book. We have limited time now. Assuming that genes at the end of life would be immune from selection, the old Medawar hypothesis. Denham-Harman. Now, again, an odd thing about that is, at what point does an enzyme switch from its Dr. Jekyll face to its Mr. Hyde face? At what point does it become deleterious? Why does it become deleterious at that point? There's just absolutely no explanation for that. It's pretty pointless. Is it a random change? Does it occur because of some other changes associated with aging, in which case it's not what it's said to be? All right, let's leave that aside. And we'll then go to a man who actually preceded George Williams. This is Denham-Harman. Denham-Harman was an engineer. He's an electrical engineer, I believe. And he believed he understood what caused aging. it wasn't some mysterious change of a protein from Dr. Jekyll to Mr. Hyde. He believed it was the accumulation of damage caused by the natural high energy particles present in the environment. He knew that these were produced externally. Cosmic radiation was a big thing at that time, as I recall. And he knew they were internally, although now we know much more about the internal action of reactive oxygen species, etc. But my only objection to that is his experiments. He tried being animals and cells in high radiation environments and found there was some sort of resemblance to natural aging, not a very good resemblance. And he didn't know that cells had natural repair systems to correct mistakes in DNA. Even some apparently immutable mistakes could be corrected with a fair degree of accuracy. Okay. Thomas Kirkwood, who is presently a well-known proponent of his own disposable aging theory and pretty much composes the editorial board of many publications devoted to aging. Not composes entirely, is on. Thomas Kirkwood's theory of disposable stoma basically is this. Nature has limited energy. Organisms in nature have limited energy, and nature cares nothing about the individual, it cares about the species. So an organism will reserve that limited energy for perfect repair of its germline cells, while its somatic cells, its body cells, are ultimately disposable. They're essentially like that fellow who wrote The Selfish Gene said, the body is basically a mule designed to carry genes. So that's the basic view, except, okay, I say that's a sort of group selection on a micro scale. The cells giving up their perfect repair to the germline cells, sacrificing their own lives for other cells. It's very clear that living animals have more than enough energy for reproduction, avoid predators, etc., including play and activities that make no sense if they don't have the energy to produce a few reparative enzymes, which don't take very much energy whatsoever. The number of genes that change with aging is very small, a very small proportion of the total genome. And he's also wrong about the perfect group of germline cells, because recent work within the last month has shown that early embryos have the same epigenetic ages as their mothers do until it's gastrulation, where there is an actual rejuvenation process, which at the very least resets their age clock. We're talking about their DNA methylation age clock as per Hannam Levine or my favorite, Steve Horvath. Sets their rejuvenation clock back to 0. Does it also repair all their damages? We know that a highly defective mother yeast cell with all kinds of lesions in her DNA will produce perfect offspring. how good is this repair process? How perfect is it? It apparently is good enough to last for generations. Further experimental work in mice and lower mammals shows that there are a whole bunch of enzymes and important factors that are reduced during aging, actually, and even more that are increased during aging, particularly among microRNAs. What point am I trying to make? What I'm trying to say is that we know that there are changes in the transcriptional and translational profile of cells with age. Transcriptional profile is basically the age phenotype of the cell. Okay. Now, David, I just put this in at the last moment because I really didn't know much about David Sinclair's theory of aging. I know David Sinclair. I was very delighted with his review of my my bio archive. I can't call it a pre-publication, preprint. But basically, he seems to be a believer in this same sort of stochastic random damage theory that everybody else believes in, except he's switched from the damages occurring to nuclear DNA, because we found just aren't damages in nuclear DNA, certainly not enough to cause the aging phenomena, to mitochondrial DNA. Yeah, there's a high source of reactive oxygen species. It should be easily mutable, but that doesn't seem to be the case either. So he has shifted to somehow a loss of epigenetic information. This is his information theory of aging. That basically genes don't change. It's the somatic mutation theory of aging, that genes don't change with aging, but the epigenetic control of genes changes or becomes lost with aging. So this is another wear and tear, except the wear and tear is on the epigenome rather than the genome. What he does have is a working plan for which he is economically involved in for treating aging, which involves rapamycin, metformin, and most notably MNN, nicotinamide mononucleotide, which is a precursor to NAD+. And the basic theory is that we keep on losing, it's a fact, we keep on losing NAD+ as we age. It starts accumulating in reduced form in the mitochondrion, and its cousin, which exists in the cytoplasm, called NADP+, is present abundantly, but it doesn't help. It doesn't help the mitochondrion. In fact, what's missing there is the reduced form NADPH, which is actually a strong reducing agent which counteracts the damage that oxygen does to a cell. Okay, where am I? So here's a very prejudiced slide. It's the pros and cons. It must be David. The pros and cons of program versus stochastic theories of aging. Pros: stochastic theories of aging, they're easy to understand. The new car to jalopy model explains it all. Do you understand what I'm saying?
Yeah, sure.
Yeah. Program theories explain species lifespans and the mechanisms that they deliberately limit them. Explain a lot more than that, as I'll in a second. For now, I'll tell you right this second. There's in all terrestrial vertebrate genera, classes rather, mammals, birds, amphibians, reptiles, there is a definite relationship between the time from conception to sexual maturity and lifespan. Now, that, according to David Fells, the naturalist, is because lifespan is, well, he says that lifespan is related to predation rate, which is statistically a fact. But he can't understand how lifespan length goes backward and influences the time period from conception to sexual maturity. But I believe that that I can. Okay. Cons, stochastic theories. Please note that none of these theories predict species maximal lifespans. There's no way to predict it if it's basically a series of random events, or how lifespan is related to ecological niche, which it is. All of these theories assume the so-called hallmarks of aging. This is a very famous paper by Louis Autain that pretty much all people in the biology of aging subscribe to. And the hallmarks of aging are very explicitly dedicated or devoted or directed towards cellular aging. So the basic theory is that cellular aging is the basis of aging. We'll talk about that in a second. None of these theories explains the correlation of time from fertilization to sexual maturity to the lifespan of the animal, or the way that predation determines lifespan and hence the length of time until sexual maturity. As will be discussed, maximum or average lifespan is a function of time to maturity. In mammals, it's a function that involves an exponential term. In other orders, in other classes of terrestrial vertebrates, it's often just a multiplication factor. You multiply the time till sexual maturity by this factor to get the total lifespan. And it works short lifespan and long lifespan. All right, I don't mean to bore you. Here's something non-boring. Quote from the Bible: "For the life of the flesh is in the blood." And alternatively, a quote from a movie I watched as a kid, Dracula, with Boris Karloff. "The blood is the life." And next to that is Nature, volume 433, 17 February 2005. I can't read it. I guess I can move you guys out of the way. Oh yeah, not hard. Rejuvenation of aged progenitor cells by exposure to a young systemic environment. Now, this is a paper that, to me, changed everything, and I believe should change the paradigm of aging completely. Let's see what the next slide looks like, and perhaps I'll put you back. It's pretty horrible. Here on the left, we're showing basically the process of parabiosis. Now, Parabiosis is a process by which two animals are wounded and stitched together in such a manner that they share a common circulatory system. I can't imagine what ethics committee would allow such an experiment to be performed. It's often the case that the older, we're interested in what's called heterochronic parabiosis, where the two rats sewn together are of different ages, one young, one old. Often the old rat will bite off the head of the young rat, killing the young rat and itself, since they have joint circulations. It's a pretty cruel procedure. It was, I can't remember, it was first thought of in the 1800s. By 1940, McKay used it to try, we used heterochronic parabiosis. Clive McKay, the guy who discovered that calorie restriction increased lifespan, he tried this procedure, heterochronic parabiosis, on young and old rats, and he reached the conclusion that that the old rats were euthanized by the parabiotic treatment, in that when he dissected them, he found that their connective tissues were white instead of yellow, and their flesh was easy to cut through instead of hard, like the flesh of older animals. If anybody's eaten an old chicken, they know what I mean. So that was not really great evidence. Ludwig, in the 1940s, tried the same experiment and discovered that he could increase the maximal lifespan of the old rats by something like 20%. Now, this is pretty amazing when you think about it, but one could easily explain it by a much simpler hypothesis. One could simply say that the functions of the old rat were taken over by the organs of the young rat, excretion by the young rat's kidneys, and etc. So it was when the Stanford group investigated parabiosis and found that the environment actually changed the aging characteristic of cells. In this case, progenitor cells, which are basically stem cells that only produce one or two sorts of offspring instead of a multitude. They have a very low potency, as they say. Suddenly, that really changed my whole idea of aging. I looked back to other experiments where they did tissue transplantations between young and old animals and et cetera, and found that while if it were the case that aging were the result of irreversible damage accumulated by a cell during the course of its lifetime, then if you took old cells and put them into a young body and asked them to form a tissue to heal a wound, you'd expect that the old cells would fail because they just don't have what it takes to reproduce quickly enough and survive the conditions of transplantation, etc. And if you took young cells and put them in an old body, why, those young cells are vigorous and full of activity, they'll certainly make a much stronger joint adhesion, wound repair, etc., than the old cells will. But in fact, the opposite is the truth. Old cells placed in a young body become active, are able to form grafts and are able to form viable transplants, while while young cells put in an old body basically become old and viable, etc. So this is later on, the Stanford group went on, the individual members thereof, Tom Rando, the Conboys, Nader Weissman, but
Amy
Wagers,
Wagers.
yes, went on to do further work in parabiosis. And she was joined apparently at Harvard by Saul Villeda, who used parabiosis to show that improved the mental functioning of old rats. They became normal in terms of their ability to. They became normal young rats in terms of their ability to learn. So later studies show that just about every reproducing tissue was rejuvenated by young plasma, or by the components of young plasma. The Conboys went a little further to show that, in fact, there was no cellular contribution by the blood cells to the new grass and etc. Anyway, so the paradigm shift is at least in part simply that instead of, pardon me while I move you, I didn't have it. Instead of the aging cell producing the aging body, that is, instead of aging resulting from cellular aging, the aging body produces the aging cell, or more accurately, it produces the cell's aging phenotype. And then the rest talks about the illustrations on the left. So what I'm saying, which is demonstrated by Conboys, by Villeda, by Wagers, by Rando, although they never say it, is that the cellular aging phenotype is mutable. It's changeable. We can bring it back. We can bring it back to the youthful phenotype, because it's controlled somehow by blood, and specifically by the blood plasma. So let me move you guys back over here and see what the next slide says. So this is my tension. A life is a four-dimensional object that unfolds over time. Well, of course, it unfolds over time. But, you know, if cellular aging is a result of accumulated damage, and if the cell age phenotype that results from accumulated damage is controlled by the body, then the damage isn't random. This to me seems very clear. That means that both life stages and the rate of duration are fuzzily determined. Now, I'll talk a little bit more about that. Cellular age phenotype is controllable by factors in the blood plasma, which means that cellular aging is a myth. There's a periodical called The Aging Cell. I say there is no aging cell, that there are cells that are extracted from old people, show all the characteristics of, quote, aging cells, including heterochromic and mitochondrial inefficiency and ROS production and different set of transcriptions, translation products. But if you take that same cell and put it in the proper environment, it will change so that it now has a phenotype appropriate to that environment. Okay. The question remains as to whether it is pro-aging factors in old blood or pro-youth factors in young blood, or a combination that results in cellular age determination. Our own experiments show pro-youth factors can reverse cellular age phenotype, including the Horvath clocks, the epigenetic age signature of cells. So it doesn't answer the question. The Conboys try to answer that question, work together with what's his name, Dobry Kiprov, by diluting plasma in half with a solution consisting of saline and off-the-shelf young serum albumin, human serum albumin. And they showed that there was apparent rejuvenation in tissues. They never showed a Horvath clot. They never told how long this rejuvenation lasted. But there did seem to be rejuvenation. I've seen it replicated by some Chinese work recently. But another Chinese work said that showed, in fact, that young serum albumin is a pro-youthful factor. This is what Dobry Kiprov originally believed. And so I would say at this point the experiment is inconclusive, showing that it's exclusively pro-aging factors that accumulate in the blood. To me, that's another extension of the wear and tear theory of aging. We know that there are youthful factors that exist in the blood that promote youth, and our experiments have shown that we have reduced age by 54%. We have reduced old rats to young rats, young adult rats, not children, not cubs, I guess they call them. Which means that cellular aging is a myth. The cell's apparent age, its age phenotype is determined by its environment, not by its history. Okay. So what is life, real life, say about aging? So I'm saying, to me, this is amazing. We're basing our theory of aging on a simplified version of Darwinism, holding that as a Bible, as a mathematically precise discipline in order to evaluate our theories of aging. I think we should evaluate our theories of evolution and leave aging to experimental verification. And that's what I've done. I've looked at those instances where aging has been reversed. And I connected a few dots. Okay, so the real answers to lifespan and aging are not to be found within the thin volume of Darwinian evolution, but in the natural world. After examining hundreds of genera and of terrestrial vertebrates, thousands of species, the eminent naturalist David Ricklefs concluded, thus, no single pattern of life, development, maturity, aging, varies among vertebrate species only by expansion or contraction of a common time scale. So I say, the meaning of this is clear. The life histories of all terrestrial vertebrates are similar in all, the shape of their lives. And at this point, I realize what I neglected. We look at human development. We see prenatal development. It goes embryo, from zygote to embryo to fetus, neonate to etc. We go to toddler to school child, preteen to adolescent, and then to adulthood, and then and then it stops as far as development is concerned. It goes from young adult to middle age and to old age. And I suppose you can divide each of those periods into early, late, and middle, but they exist. And the fact that they exist is that we can recognize them. And the fact that such phenomena as pattern occurs means that these genes are manifesting themselves after reproductive life, but that's all right. The point is that all mammals basically have the same life, to sexual maturity, then finding a mate, raising a family, or taking care of your herd, depending on whether you're male or female. Eventually giving into old age and allowing the strongest competitor to defeat you, handing over your role, as it were. We all have the same pattern. The only difference is the rate at which it occurs. In a mouse, all the stages occur in the space of 3 to 4 years. In a human, all the stages occur in the space of 120 years, but as we age, our probability of death increases exponentially, so that very, very few of us ever reach the maximal possibility of our lifespan. Though we do see at the very end of lifespan, those individuals who make it to the very maximal seem to stop aging. We see this all the time. Nobody has an explanation. It's just that the aging program has run out. There's nowhere to go. At that point, we just continue as we are. Okay. The biologist, and apologize for mispronouncing his name, João Pedro de Magalhães. I've written to him, though a firm believer, I've communicated with him, in the modern synthesis, states that after examining the literature of terrestrial species, and he did a meta-analysis involving lots and lots of papers, overall, these results indicate that independently of body size, the developmental time is strongly associated with maximum adult lifespan. Developmental time, the time to sexual maturity, is strongly associated with maximum adult lifespan. And by the way, maximum adult lifespan is strongly associated with the average adult lifespan. It's a fraction. Okay, now how do I go back? I didn't mean to do this. Now that I'm here, let's go here. So what I tried to do, and what I told people in my my 2013 paper, was what I called heterochronic plasma exchange, HPE. I figured that was nice commercially because it could spell hope or it could spell hype, depending on your take. But the idea was simply this: whether the convoys are correct or not, whether it's a case of both youth and promote factors and age promoting factors, if you completely replace the plasma of an old person with the plasma of a young person, you should be able to change that young person's cell's age phenotype. I figured test, if you did it enough times, you could do it, it would permanently revert. Now, fortunately, we were not able to do that. We tried every which way. We had people in Germany who supposedly had done that, had done plasma exchange with rats. We picked rats because they have relatively large veins. Mice are just too small. So we tried everything, and we even tried manual plasma exchange, extract blood, centrifuge it down, exchange it with young plasma, re-inject it. The rats' veins wouldn't take such abusive treatment. So at that point, this was in Mumbai, India, at NMMIMS University, which is a known school of pharmacology and a known school of business. In any case, I only had e-visas at that time, which gave me 2 months stay, and I had to leave. And I knew if I left, I'd be leaving without results, I'd probably not be coming back. And honestly, that was kind of a relief. But in any way, I said, let's try it. Let's try one last thing. I really read all the literature I could on the subject. I'm not sure of what to say. I connected a few dots that to me were very obvious and apparent, but nobody had ever mentioned them except in passing. And I said, I think I know how to extract that fraction of blood which contains what we now call E5, the youth promoting fraction of blood. Now it was a guess. It was a guess and 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 a guess, all together, each one with a small probability. So when I got home from India, it was really good to see my wife and family, etc. And I honestly expected that was it. Akshay will say, well, we tried. Akshay, by the way, sponsors all of this and is a financier and a biologist in his own right, although self-taught. And I was not totally unhappy because at that time I was 72 years old and or more, and traveling was very difficult for me. Just walking the gangplank, just walking in line through customs was like a real effort. For every foot I had to move was painful. Every step, rather. But I wanted to see it happen anyway. So just telling you I was pleased about not going. All right, if it doesn't work, it doesn't work. What can I do? I tried my best. But then I got an email from Kavita that it works. And this was our first try. So what I had done is we extracted that fraction of plasma that I thought would contain the pro-youthful factors. I can't do much more than that except injected it into rats, into their tail veins. Strada's really excellent at that. She is really excellent. Thank my laboratory skills are somewhat lacking, but not my inventiveness in the laboratory, at least. So that's good. So here we can see, we look at the first graph and we see grip strength. This is in newtons. A newton's about a quarter of a pound. So on day, we have blue represents the old control. The middle represents the young, the old treated, and the gray bar represents the young control. And actually do see a slight increase in strength in the young control, but we see an enormous increase in strength in the old treated animal, to the extent that at one point it even surpasses the young control, and certainly overlaps the young controls if you look at the error bars, and no overlap whatever with the old control. So this was a little bit encouraging, and actually forced me to go back to India. The next slide shows this is a normal human blood panel, except a rat. So we look at bilirubin, a measure of the gallbladder, or the destruction of hemoglobin, gall. We look at liver enzymes, we look at triglycerides, we look at high density lipoproteins, cholesterol, glucose, creatine, blood urea nitrogen. That's what that stands for. It's a function of, that's a variable fit kidney functioning, and total protein. And we see in almost every case, not in almost every case, in every case, that the old experimental animal becomes just like the young control, which again, reemphasizes the nature of life as a transition of life stages. They have reassumed the phenotype appropriate to their epigenetic age, as far as I'm concerned. Or, you can read the book, read the original paper. There's much more on this. For instance, for me, what's particularly important is the amount of reduced glutathione present in organs. Glutathione is an antioxidant. It's very, very important in combating oxygen damage due to mitochondrial respiration. What we see is just that we get back in all aspects to a life stage resembling or identical to that of youth in every aspect tested. And these are a lot of different tests. We have many more tests, and of course, the paper in the book shows tests, cognitive tests. Let's see. Our second try shows that. With our first try, we sacrifice the animals after 30 days. With our second try, we're going to do the same thing to repeat it, and said, no, let these animals live, at least for another 30 days. And so 1 month, 2 months, and you can see what you're looking at here is days post-treatment on the horizontal axis, and latency, quote, on the vertical axis. Latency means the time it takes for the rat to solve a maze. This is called a Barnes maze. It's just a big table with circular holes cut out along the periphery, one of which has a pouch, rest of which lead to a big drop to the floor, which rats won't take. So they search around for the one with the pouch, and we give them visual cues. In any case, we can see a continual improvement in the rats. I think what we see over here in the third month, and this is past second injection. The second injection at day 95. Well, let me see. The second injection at day 95. Oh, I think I have to move you guys. Yes. So the third month is past their second rejuvenation set. It's a set of 4 injections. Injections, nothing more than that. And we can see that at time 0, where originally the old rats would just sit there in the middle of the table with a confused look on their faces, they kind of got the idea much more quickly to try to find their way to a hole. They're also much larger, by the way. These are male rats. Male rats grow to be about the 18-month-old rats were about twice the size of the 5-month-old rats. So, any case, there's a lot more to this can try, as you see. Some very exciting things, like, for instance, cytokines being repressed to below youthful levels. And lastly, I just want to conclude by saying, due to the beneficence of B.A. Clonel of Belgium, we were given some money to do longevity experiments. Longevity wasn't our aim because the FDA couldn't care about longevity, and we want to become commercial. So, for instance, the increase in grip strength is a good indication that maybe frailty should be our first target, as it's a low-hanging fruit. It's absolutely universal, and there's absolutely no cure for it. But so this is the little part of our longevity study, and let me see if. Okay. What the heck? Oh, stupid. I forgot. How do I go backwards?
Press the left arrow, perhaps.
Left arrow.
And the cursor keys. Or you can click on the bottom left there. You can go back. The video's running now.
Yeah.
You went too far, so go back. Press the left arrow.
I went to, let's see this one, because the other one is unlabeled. This one is labeled. And I wanted everyone to see this. One, we see that after the third dose, the grip strength remains at a high level, at least twice that of the old control. And I wanted you to see that there was basically no change in body weight. This is something that bothered Akshay. He thought that young rats should be slim. I said, well, why should the growth rate change? I think Akshay was right in this case, because towards the end of the experiment, after the third dose, we begin to see that, one, the body weight decreases only slightly, but decreases while the strength increases considerably. And we can see that, by comparing these two. And what might the result of that be? And that's the next slide. That's the next slide. I'm not on. Let's just click on it, and I should.
If you click on the bit from current slide at the top. top, it's the
From
second
current
one. That's the one,
slide.
yes.
Ah,
And then
Now,
click on the button
this
there.
is, oh, no, I understand that. This is, I just wanted to point out first that the C stands for control and the T stands for treated. And even this still photo, I think you can see the difference between the two animals. The treated animal is longer and sleeker. Now, I must admit that this is a particularly good example. And in at least one case, the control seemed better than, but this is a normal example. This is the more usual and typical example of treated versus controlled rats. They're put in the same box just for display. So let's watch their video and see if we can see a difference. And that's about it, folks.
So we saw the treated rat with much more vigor and much more vitality in that particular case.
I would say so. Also slimmer and sexier, if you're into rats.
But there are many other metrics as well.
Shapelier. And there are many other metrics as well. Yes.
Yes. So how's anybody?
Non-invasive metrics, of course. Our choice of inflammatory cytokines and such like things that you can easily measure in the blood.
So the actual plasma fraction, E5, is commercially sensitive, and so you're not publishing for the time being the details of that.
For the time being, yes.
But would it be possible for somebody else to try and repeat the experiments that you and your team have done?
We
Because
plan to.
that's what science is about, isn't it?
Course,
science is about other people replicating.
Exactly. I would not trust anything unless it could be replicated. And it isn't science unless it can be replicated. And I don't really blame people for holding off on their judgment until we've replicated it. But because we feel certain we can, we say that. We certainly have intentions in the future of human, dog, non-human primate, and further CRO, a certified research organization or commercial research organization, certifying all of our results. Some of our results actually are already certified in our papers. A lot of our organ levels of antioxidants are determined, were determined by private labs.
And how do you envision the tests working in the case of humans? When the rats are being injected, every 90 days? So with humans, how would it work, and what effects do you imagine you'll see?
I mean, that's of course is the question. What we have seen is what I would call the Benjamin Button effect, which is that the older the rats get, the younger they appear, or the younger their biological age. So it takes time for the effect to occur. How much time? I can't say. Akshay is the optimist. He thinks it'll take as long as it does on rats. I'm optimistic in a different way. I believe it will reset the age, especially after the second set of rejuvenations. It'll reset the age eventually, and it may take years to the young adult stage. You'll be growing younger instead of older, is how I envision it. And
because it might take years.
People complain that it might take years. And I said, would you really mind growing younger every day for years?
Because it might take a long time in the human case, I can see it makes sense to try some intermediate species such as dogs. So what timescales do you have in mind when some of these other experiments might be done? And what's the constraint? What's limiting more people doing more work on this?
The limitation at this point, which we hope won't be a limitation for very long, is amount.
Amount of E5.
Yes. Our major purpose in the United States is to increase to useful values for people, scaling up, as it were.
So some people may imagine that you get E5 by blood transfusions from young people and removing stuff. Other people may imagine you create them all from a small number of biochemicals.
I really don't want to talk about that. Sorry.
That's fair. I thought I would ask.
No, fair enough. You can always ask.
Right. Okay. And we're actually already going 90 minutes. So normally I would shut down these meetings by now, but you had such a lot of interesting things that I let you keep talking. So maybe we'll keep going in questions for a bit longer. I will pick out some questions from the audience too, if the audience members upvote to help me pick out which of the questions are more appropriate than others. In the meantime, I will ask one more myself, which is, you had this nice slide of two different theories of aging, the stochastic theories of aging versus the program theory of age. Basically, you believe that somehow for the good of the species, as it were, there is a program that applies which will cause us to eventually grow old and leave space for our descendants who might vary from us. And you said, well, one of the drawbacks of the program theory, and you said, well, there are none. But I guess people will ask about two drawbacks. They'll ask, first of all, well, where does this program operate? Can they see the program?
No. I don't know. We don't know.
So that makes it still a bit of a mystery, because people can see the damage accumulating.
Again, I've said before, I think we've uncovered a new continent, and we've seen some of the taller peaks, but there's a vast amount to yet be explored. Yeah, I don't know where it comes from. Is it the hypothalamus, the suprachiasmatic nucleus that regulates everything? I don't know.
But you think it's to do with the blood in particular? The blood is.
I know it's to do with the blood.
So why aren't more people in the rejuvenation community jumping up and down about this? Why aren't they saying this is it because of this?
Because they don't believe it. I think it's that simple.
They don't believe the experiments will be repeated, or they don't believe the theories that you have advocated?
I don't think anybody's heard those theories until very recently. I got some very positive feedback from eminent theorists on it. So including, can I mention his name, Nicholas? Yes, Steve Horvath, the inventor of the Horvath clock, very well-known science, and from many people I've gotten. So the theory, I believe, is correct. I haven't heard anybody offer any logical objection to it or to anything I've said.
Well, the other logical objection is on the notion of group selection, because people will say, if something's for the good of the group, if animals have to behave in some kind of self-sacrificing way for the group to benefit, what prevents free riders? You know, this is the guy who benefits from the group as a whole, but that guy is more selfish than the others. What's going to prevent the evolution of that individual trait and damage the group trait? And this is the classic argument against group selection.
Yes, correct. That's against group selection as classically conceived. But group selection, by my broader definition, is the elimination of an entire species by, for instance, invading species, climatic conditions, etc., where whole groups replace other groups based on their superiority under changing conditions. So I have no argument with that sort of Talmudic logic.
But people will say that it might be for the good for the species that all individuals grow old and die, but if there was a genetic variation that allowed them to not grow old and die, then, and they pass that on to their descendants, then that
Correct.
would dominate. And so the ones that grew
Correct,
old and die
correct.
would die, and the ones who lived longer would live longer. So,
But the fact is that the population lifespan is largely determined by predation. So that argument really doesn't hold when you throw predation into the picture. If they're going to be eaten, they're going to be eaten.
I guess there's the other argument.
Doesn't matter whether they'll live to be 1,000 or not, if they'll be eaten by the time they're 2.
There's the other argument that a group that can also fight against free riders, as we humans do. We've got sensibilities to not tolerate selfish individuals in our midst. And
Yes.
this seems to apply up and down the biological level too, that group selection can still make sense, provided these other mechanisms are in place at the same time.
That's correct. Nobody seems to consider the fact that often organisms cooperate with each other to produce a common effect that's greater than the organisms by themselves. Like, for instance, wrasse fish that clean other fish. By the way, those are you know the cleaner wrasse, WRASSE. They follow other fish and pick the parasites off them. For example, other fish come to their cleaning stations to be cleaned. In fact, so there are cooperations in nature too. It's not all red in tooth and claw.
A question from Didier Cornell. He actually has a few questions. First is, why don't you give the information to everybody? If it's working, people will give you lots of money and fame.
I have an obligation to Akshay and the shareholders and everybody I promised. I keep my promises. That's the way it is.
So in that case, Didier's next question is, well, how long until the patent will be available and you are able to talk? Is this years, decades?
Oh, God forbid. I'm 77 years old. I have my own shelf life. I want to get it done as soon as possible, but I have no idea. Let Didier speak to in the next 2 years. Maybe.
Okay. People have questions about other ways in which the body might be prompted to rejuvenate itself. For example, Brian from Canada talks about his own experience with water fasts. And he says it's only N equals 1, but there does seem to be some literature that the body can be moved.
Absolutely correct. Absolutely been shown that epigenetic age is changed by exercise and diet.
So the effects that you expect from E5, how will they compare with the effects of what we can do by diet and by exercise and so on?
You know, I'm 77 years old. I'm still pretty strong. I actually went to the gym yesterday, but I can walk 15 minutes and I'm dead. I remember when I thought that I could walk forever. I used to walk from Lower Manhattan to the Bronx, where I lived. To get back that energy and not to mention all the sensory deprivation that occurs. My hearing is poor. My vision is actually better than it's ever been since I had my my lenses replaced with artificial lenses. But
so these other mechanisms can give some rejuvenation, but limited.
no, we plan. You know, our rejuvenation occurs at the cellular level, at the heart of the cell, at the cell's DNA, at the epigenetic level. I mean, this is, for instance, what David Sinclair is trying to do, reprogramming. As far as we're concerned, we discovered nature's way of reprogramming cells.
David Sinclair's looking at a different mechanism using 3 of the 4 Yamanaka factors.
Correct.
OKS, so-called,
Yes.
which seem to be remarkable. But as you said, maybe it's not what nature has already provided.
Right. We still to see it in whole organism. We still to see if it prevents teratoma formation, etc. And ultimately, our way is better.
Yes. I'm going to take one more question just now from the audience. Then I'm going to pass the microphone after you've answered it, Carol. I'll pass it to Nicholas and Nina because they've been listening very thoughtfully. I'm sure they've got some comments or questions, but let's take this question first from Steve Buss, who is an aging researcher who says that the articles you published in 2015 or so are remarkable and changed his own perspective. So he's been following you for a while.
Thank you.
He asks a fairly technical question. He talks about JMJD3, a demethylator of another big chemical, H3K27Me3. So I'm not sure.
H3K27Me3 is the marker of the polycomb restriction apparatus.
No.
That's one of the amino acid residues in the histone proteins that DNA is wrapped around. And when that particular configuration exists, that means that this DNA should not be touched, should not be fixed, should not be transcribed. It's kind of a stop signal. It's a repressive signal. But go ahead.
So I'm glad you know what that means, because Steve says, wouldn't a good way to achieve the rejuvenation you talk about be to increase JMJD3, this demethylator? And he says there are various things you can do.
Try it.
So he can try it, but you've already found something that is working, and so
we're not going to go off in different directions at this point. we're pretty focused.
So there's a research program there that you are basically supportive of, but you're not going to do it yourself because you need to focus. Absolutely.
Yeah, exactly.
So Nicholas and Nina, the publishers of Harold's book, what comments would you like to add or questions would you like to pose to Harold?
Well, I would like to comment about, you asked why aren't other people from the rejuvenation field as excited about this matter? And I think one of the matters is that it is a paradigm shift, and all paradigm shifts are hard and people tend to take a long time to jump on board. And
typically they say the older generation has to die out before the new paradigm gains acceptance. I can't wait for that.
Let's hope that doesn't happen. And another thing is that Harold defends the programmed aging paradigm. And this is something that a lot of people, I feel personally, this is my opinion, people are kind of afraid of thinking about that, because if aging is programmed, then you can change it. But Harold's trying to show that even if it's programmed, it can be changed.
Yeah. And thank you, Nina. That's a point I make in the book. It's one of my few witticisms. basically, people have been afraid to look at programmed aging. That's originally what stopped August Weismann's theory back at the turn of the 20th century. If aging is programmed, if life is a program, then your death is inevitable. Sometimes the best thing to do is to confront your worst fears, and you often find solutions when you do so. And that's really what I did, because the fact that lifespan is programmed, I think, is obvious to anybody who looks at animal lifespans in the natural world. But just the very concept, oh my gosh, our death is fixed, immutable. We're four-dimensional objects with a beginning and an end is just too scary to contemplate. But once you do contemplate it and realize the mechanisms behind it, it can actually be changed.
Yeah. And another thing, that people aren't 100% on board. I'm also not 100% on board. I believe in Harold. I believe in his experiments. But as anyone who knows science, there needs to be more experiments, more results, and everything else. So I'm excited about this research. I support Harold. I think he's doing a great job. And we also published his book. It's also a way to show the support. But I'm also a bit well, let's see what happens. Let's see the next experiments.
We certainly don't intend to end here.
Yeah.
We don't intend. Okay.
And yes, regarding what Didier asked about why, because you could open the information for the whole world, like Didier suggested, open the E5 formula or something like that. And no, but regarding that, you said that you have you keep your promises, but I think it's not just that.
It's not just that.
Yeah, it's not just that. You are in a very very hard situation because you was looking for help in the rejuvenation space and nobody really gave you the material conditions until Akshay appears. So
yes,
we can't back on time now. Back on time and help you some years ago. So Akshay already helped, and he took many risks, and the investors
invested his own personal money, and his friends are investors.
So what I would suggest to Didier and everybody in the rejuvenation field is that we work with what we have, the actual situation. We can't change the past. So now what we have Harold and Akshay working together with investors, and many great inventions in humanity happened with this way, these cell phones, computers, all was made with patents, with companies. Okay, so we need to work with what we have. We can't change the past. So let's help in the way we can. So everybody, Didier is helping, us, me and Nina, we are helping, but we help in the way we can, because we can't change the past. So that's the comment I would like to make.
One example here, from the field of flight. The Wright brothers are famous for having flown first in December 1903, but they were generally disbelieved or widely disbelieved until they put on public flying exhibitions in Paris and in America in 1908. So they went in a period of 5 years in which they were not widely believed. The newspaper headline said, well, have the rights flown or have they not flown? After all, there was this photo, but people said, well, it's just bouncing along the beach, you know, a bit like the Maharishis sometimes seem to be flying in meditation, but actually they're just bouncing off the cushions. The reason the rights were slow is they were doing exactly what Harold is doing now. They were securing patents and securing legal agreements, securing customer relations. So they talked a lot about what they were doing. They did discuss, but the final vindication had to wait until patents were in place. So I hope it doesn't take 5 years in this case. I hope it can be done more quickly. But in any case, something else might make people pay more attention, and that is the results of your ongoing test in terms of how long might some of these rats live, because I understand that the rats of this species, generally the maximum recorded lifespan is somewhere about 4 years. Other rats from other species may have lived longer in some, but this particular species, I forget the name. So how old are the rats now, and how long would we have to wait? If
the sprig dolly rats, they're only about 32 months old, so we're going to have to wait for more than a year to. But during that time, we're talking about maximum length. During that time, we expect the old, God, I don't want to hope for death for any of these poor rats. We hope that the old controls die while the treated rats remain alive, and that'll be a good indication that we're heading towards the ultimate prize, which is immortal youth.
So in a moment, I'm going to close down this formal part of the session. Just for people still watching, almost all of the audience is still here, so that's very encouraging. I'm going to let you have a chance, Harold, to make some closing remarks. If there's something you feel deserves to be emphasized again, then you can do that. And then I will break for 10 minutes. I will shut down the recording. I will shut off the cameras. And then if people do want to come back, a few of us will still be here, and we can continue to discuss in a more informal way. And people who have been asking me, could they raise a question by audio? I will give them a chance to do that now. So whether Harold and Nicholas and Nina are still here, it depends on their schedule. They've already given a lot of their time. But I want to thank you, Harold. You've given a great deal for people to think about, and it's only the tip of the iceberg. There's a lot more in your book, which is not a book that may be for everybody's liking, because you are discursive. You look at multiple lines of science and philosophy and history, which I personally love, because I think all of that's important. Other people may just want to skip to the end where you give a very powerful summary of the data so far. But what would you like to say in conclusion of this part of the meeting, Harold?
My hope is that this will be an evolutionary change in mankind for the better, that people will no longer look at the future as something to hand on to their imaginary grandchildren, descendants, but as their own future, and will take care of the earth. People will be less likely to throw away their lives in the useless ego struggles of dictators when their lives could be so productive and last so long. And my real reason for even thinking about it, because I personally don't think life is all that wonderful. It is wonderful, but my real reason for wanting to extend it is that it allows human beings to have a galactic civilization, to reach the stars, because the speed of light is still, in spite of those UAPs that the Air Force talks about, the speed of light is, to our knowledge, still the limiting velocity in the universe, and it takes years or decades to reach the nearest habitable stars.
So this changes everything.
Yeah, I think
it's not just a small matter. Oh, a little bit different in this weird part of life. No, this will change philosophy, change religion, change society. And when people find out that it's for real, rather than just a pipe dream, they'll stop doing what they're doing. They'll stop being distracted, and I think there'll be a much quicker change, almost a singularity in human experience. So thank you very much. It's been a real pleasure to host you, and thanks also to Nicholas and Nina for joining in.
Thank you.
Yes, of course. Thank
Okay.
you.
Thank
Bye-bye.
Bye-bye. This